It's a real pleasure to be here. Thank you for the invitation. I know that this is a neurodevelopment preclinical symposium. I'm not going to talk about a neurodevelopmental model at all. I worked in this space for a long time, and I kind of got a little disheartened with my work, sort of considering, well, you can do a lot of things to alter the development of the brain in a rodent or another model. But when we get to an adult, what are we trying to test? What's going to be relevant from that species to humans? And that's kind of what I want to talk to you today about. What the work I've been doing using this decision-making is something that we can test in rodents and primates, as well as people, and hopefully as closely as possible. Thankfully, both the speakers have introduced a lot of my background for me, so I can hopefully catch up a few minutes here and I get straight to the data. So I'd like to thank everyone who's involved. I don't work in a silo. I test people and animals, so I need clinicians as well as basic researchers. So a lot of acknowledgements to give. I'd like to get out first. So this is the space I live in. We need to use rodents because we have tools to understand mechanisms. What does the brain do? How does it compute things in our daily lives? How do we make choices based on those computations? We can't get into the brain of a person the same way we can get into the brain of a mouse. And that's where we can understand sort of what those fundamental processes are. But the real key is to try and understand how we can help people having decision-making problems, be that via psychiatric disorders like schizophrenia and psychosis, or any other problem in their daily lives. So really, what I do is try to match phenotypes. We see a behavioral change in a group of people. What do we need to do to the brain of a mouse to try and mimic that behavioral change? And to do that, we need to trust that the processes we're comparing are the same. So I focus on the dopaminergic system, and Tersh has introduced that nicely, as well as Melissa. But not everyone in schizophrenia, I'll say, has too much dopamine. It's only some people, obviously there's a lot of clinical data behind this, tend to have too much dopamine that might lead to their psychotic symptoms. And it's quite a strong link, but not everyone, every case is different. And the reason I focus on here, we're looking at primates on the left and humans. This is the striatum where dopamine projects to and signals in subcortical spaces and in the rodent. And we've got anatomical names, which I won't bore you with. We've also got these colored areas, which is what I focus on. And the yellow area here is where the patient data, people with psychosis tend to have too much dopamine being released into this area. It's called the associative striatum because the associative cortices come and talk to that area. And they're very important in our ability to think and perform decision-making tasks. Thankfully, rodents have a very similar area where very similar cortical areas project to. And that's the area I focus on. And there's a wealth of data in the clinical studies. This is just one example. That middle bar there is comparing age-matched controls with people with psychosis, showing that too much dopamine synthesis in the dorsomedial striatum or the associative striatum. So this is kind of like the neurobiological low-hanging fruit. We know that if you put too much dopamine into any healthy brain here, you give amphetamine to anyone, we're going to have psychosis. It's going to relate to too much dopamine in that area of the brain. So it's an easy thing we can do in a mouse to mimic what we understand is happening in the brain. So really my question started with this: how do we test for psychosis in a mouse? It sounds simple. It's not. We have to presume they don't get psychosis. We have no way of asking them. A lot of my students have tried. So, you know, it's simple to sort of see from a clinical phenotype in a person when they tell you they see something in the sky, or they're hearing voices, or they're believing something that's obviously not true. But in mice, we can't do that. We need something objective. So rather than look at how do we assess psychosis in a mouse, I look at how do we assess increased dorsomedial striatal dopamine in a mouse. If we increase dopamine in this one area of the brain of a mouse, what do we expect to see as our phenotype? So I use two tasks. I'll briefly summarize some of the results we got with one, but I'm going to focus on reversal learning, which is excellent since it's been introduced. So thank you for that. I'm going to delve a little bit deeper into it because it's my task of choice. And the reason I'm interested in these two tasks is because we know that we're looking at rodent brains here, but there are very equivalent regions in the human brain, albeit with some contention, as has been mentioned. The reason is all these very important cortical structures project to our subcortical areas. And that DMS, I'm going to refer to it a lot, the dorsomedial striatum, is where too much dopamine we think underlies psychosis in a lot of individuals. The important thing is the projections come in, our cortex does these computations, it comes in, it talks to our dorsomedial striatum, which is then involved in helping us make a choice, optimize the strategy we approach the environment with, and eventually act upon that. And we can test these things in mice. They're very smart, they can do a lot of cool stuff, just like we can. So a quick summary, some of the work we've done with goal-directed action. I'm not going to explain this task to you, but essentially when an outcome changes, do you stop selecting it if it's worse or select it more if it's better? we've shown that driving DMS dopamine in the mouse can impair goal-directed behavior. And this is similar to what people have shown in people with persistent psychosis, and same with us. And this is not all people with persistent psychosis. A lot of the people we test do just as well as all the age-matched controls, but there are a large portion that have problems with this. We've shown it in both mice and people. It gets more complex when you start adding in D2 and D1 receptor blockade, but I'm not going to go into that. But I think it's really nice that we can start to mimic some of these phenotypes. But 15 minutes, so let's go. Reversal learning. Very simply put, for a person, we would have two pictures. This has been described before. One of them is going to reward you more often than the other, and you learn to select it. For a mouse, we use little levers, and they can press the lever, and they get some strawberry milk if they're right. After a certain amount of time, and we deem as the experimenter that we think you've learned which association is better, without telling the participant, it swaps. So all of a sudden, you're not getting rewards. You hold on. And you might sample the other option and eventually learn that it's the better one. And we do this over and over, back and forth, back and forth, back and forth. So you constantly have to be paying attention to the environment, adapting your decision-making. And our little mice can do that very well as well. So you could look at one sort of global measure as, well, how fast did you get to that next criteria? How quickly did you complete that reversal? It's quite a gross measure because there's lots of ways you can get to the same endpoint. So we also look at some of the strategies people and mice use. And the simple ones I'll explain today, we go really deep into this, but just to keep it simple, is win, stay, and lose shift, kind of like reward learning and loss learning. And win, stay, essentially, if you chose the triangle and I said you've won $1, you choose the triangle again. All right, it's pretty straightforward. Now, you might not do that forever because maybe you win $2 on the alternative, but a lot of the time, people and mice will select the same option. Now, lose shift is self-explanatory. If you lost on the triangle, you might select the square instead. All right, these are two strategies that you can use pretty equivalently. And if you have a very simple reversal learning paradigm, you only need to do these two strategies. So if we were to have a protocol where if you choose a triangle, you always win, and if you choose the square, you always lose. If you always win, stay, and always lose shift, you're going to do so well, your data is useless to me, and I don't need to do it anymore. So we make it harder. We make it probabilistic. So sometimes you're not going to win if you choose the correct one. And sometimes on the poor one, you're going to win anyway. And you've got to navigate, was that real? Was that reward? Was it informative or was it misleading? And try and figure out what's going on alongside that. Makes it harder, means you do more trials, means I can analyze data better. And then we thought, well, why stop at 80-20, which is a pretty common sort of theme, where basically you win 8 out of 10 times on the good one, and you win 2 out of 10 times on the bad one. It's a big gap, though. It's quite simple once you get used to it. So we trial a whole bunch. We do 80-20, we do 80-40, 70-30. What we're doing is making the environment more uncertain. How do you respond when you're not quite sure which is better and how to keep up with that? So first, we're going to walk you through some of the data we've got in people with and without psychosis, and then walk you through some of the stuff we've done in mice to try and figure out what might underlie those processes. So first, people with early psychosis, we've only got a smaller cohort here. We find that in general, the trials it takes to do a reversal is no different between age-matched controls and people with early psychosis. And this is at 80-20 and 80-40, so slightly more uncertain environment, but no difference. They do it just as well. When we look at the strategies, as with others, we found not much difference in the strategies people with early psychosis use. So their win stay is pretty high. Their lose shift for most people, it hovers around 0.5 for probability, what we expect. But when we looked at 80-40, and this is within the same session, within the same people, all of a sudden, when it switches to a more uncertain environment, people with early psychosis tended to start lose shifting less. They responded to loss differently. Now, not many people in the literature actually test these more uncertain environments, so we weren't really expecting this. And I found it really interesting from a scientific perspective. Like if you scale the uncertainty, make this more difficult, you start to see phenotypes potentially emerging. We also had a cohort we had persistent psychosis, so I had ongoing psychosis for a period of time. We're going to look at that because there's a lot of literature in this, and we sort of replicated that alongside that. What we found is that in terms of how many trials to reach criterion, you can see here there's quite a bit of overlap. There's a little bit of an increase in that first reversal. But overall, they do, relatively similar. When we tested these same individuals with goal-directed action, as I summarized before, we actually found that these deficits, these increased trials to criterion, were more obvious in those who also had problems with their goal-directed behavior. So it seems to be sort of a subgroup of people who have more problems with their decision-making capacity. However, what was really interesting is when we looked at the strategies in this group of people. Unlike with early psychosis, people with persistent psychosis, and we're not the first to show this, tend to win stay less, but they lose shift the same. So we're looking at a reward learning deficit here. So something changes from that early psychosis stage to a persistent psychosis where 5 minutes, well, we're going fast, aren't we? All right, we'll kick on. Anyway, yeah, so different phenotype. My style, let's move on. So we're asking, what can we do? You've heard about amphetamine. We can give amphetamine to a person and it gives them psychosis if you have high enough dose. So that's what we start with mice. So we looked at how acute amphetamine would mimic psychosis in these tasks. We also looked at some neuronal activation. Suffice to say, we can use cool tools and we can look at which cells were active during behavior. But we'll get to the data. There we go. So what we found was that acute amphetamine did not alter their ability to do reversals on a gross level, but specifically affected the lose shift, like we saw in people with early psychosis. So that acute one hit of dopamine seemed to produce a similar phenotype. And underlying this was selective increased activation of cells in the dorsomedial stratum, suggesting that's the area where the cells are overactive, causing this phenotype, which links back to that sort of psychosis neurobiology that we understand. This is from a lot of cells per mouse. We can do a lot of big data with this. But we're injecting amphetamine systemically. The whole brain is getting flooded. So we want to be more specific. We can go to viral tools to just target certain neurons and activate them during behavior. So here we used a pathway-specific virus where we can activate just the projection from the substantia nigra to the dorsomedial stratum and activate that during behavior to try and mimic what too much dopamine release in just that one area would be. So we did a lot of stuff. That looks cool. Reverse learning in mice. We did chronically. So here we're looking at within subject, each mouse got chronically activated aura control where they didn't activate any neurons. And we can see at 80, on the left, they got 80-20, then we go down more uncertain environments during chronic. And this is percentage of baseline, so decrease here is worse reversal learning performance. And you can see in these more uncertain environments, chronically stimulating these neurons leads to poorer performance. When we looked at the strategies, we found no difference in the win-stay use though, even chronically overactive in these systems. We did see that same decrease in lose shift, not at all the uncertainty levels. So it's dependent on the sort of environment that these mice are making these decisions in. So no change in win-stay. Well, we're chronically activating the system. So our question was, well, what do you have to do to sort of impair reward learning in the brain? So we thought, well, let's go the alternative route. Let's actually inhibit the dorsomedial stratum. So where those cells are signaling to, let's turn them off and see what happens. And we use inhibitory viruses for this. Same process, we're still doing this analysis. We found is that you get specific reversal learning performances. So lower here is they have a harder time doing reversals at certain environments. So 80-40 is quite high rewards, but it's uncertain, more uncertain than 80-20. We see a specific decrease in reversal learning performance here. What was really interesting is when we looked at strategies, now we see decrease in win-stay use. So driving dopamine into the system seems to mimic what we see in early psychosis, whereas inhibiting the signaling of that dopamine seems to mimic more of what we see in people with persistent psychosis and no difference in lose shift use. Now I'm going to finish early probably. So overall, had to rush through this, but I hope that I got some messages across. Essentially, DMS dysfunction, which we associate with psychosis in individuals with schizophrenia, so either overactivating or inhibiting it in mice induces numerous decision-making phenotypes that we've also observed in people with psychosis. And for me, what I like about this is it's actually based on the neurobiology of what we know about underlying psychotic symptoms in people. Because a lot of people think that the psychotic symptoms are completely separate from the cognitive problems we see in people with psychosis, that they're completely two unrelated events. I don't think necessarily that's the case. I think there might be ways where if we can improve one or the other, we can help more symptoms in people who are struggling with these problems. And I hope I've shown you this is a translationally relevant approach. Mice do these tasks pretty similarly the way we do. We have to train them for a lot longer, to be fair, but they can do it and they use the same strategies and they approach it the same way. We can apply the same computational models to the data as well. So even though they have a different shaped brain and they're a lot smaller, they can do these tasks because every animal is trying to chase rewards, trying to get good outcomes and trying to learn the best way to optimize those outcomes. So decision-making in psychosis or after DMS dysfunction in a mouse seems to be also dependent on the environmental uncertainty. We saw that we didn't just see the same phenotype in people with early psychosis at 80-20 as 80-40. Now, mouse models, we see, depending on the level of certainty in the environment, different effects of the different phenotypes we use. So, I mean, for me, when I tell the researchers, I say, you've got to manipulate the uncertainty of your reversal learning tasks, because it's kind of like a dose response of a drug. Sometimes you'll see things at certain times, but not at other times. You need to test them all. And what does it mean for our understanding of psychosis and schizophrenia? Well, it's obviously speculation, but our thoughts are that maybe with reward-related learning, essentially in early psychosis, when that first psychotic episode happens, there's this, struggle of the brain to try and compute things properly. And the cortex starts working extra hard so we can maintain our decision-making, because at the base level of things, being able to do things is core to the brain. But over time, potentially, that becomes harder and harder because, your brain's working harder. So I think this is really interesting because I think it provides a plausible explanation for why, if we come in with the right services and support early, we can hopefully stall the trajectory or decline in some individuals. And obviously, as I've said, not every individual has problems with decision-making, but anyone who might end up having problems with decision-making, if we can prevent or delay that or stall it, I think that's a really good outcome. And hopefully that's where this work will eventually lead to sort of some productivity in the space of how we help people with the disorder. And that's it from me. So thank you for listening.